Well, thanks. Thanks, everybody for waiting. I think we'll get underway. First of all, on behalf of the Centre of Excellence for Plant Success in Nature and Agriculture, I'd like to acknowledge the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise, we recognise their valuable contributions to Australian and global society. And as plant scientists, we acknowledge the long tradition that they've had and long cultural connection they've had with Australian, with the Australian plant community, with the Australian environment, and their collections of seeds that will have a long-lasting effect on our community. My name's Christine Beveridge, and I'm the director of this here at the Centre for Plant Success. And it's my pleasure to introduce Dr. Alex Wu. Thanks for joining us, Alex. Alex is a crop modeller. He's well known for his research on modelling leaf photosynthetic carbon dioxide assimilation and field crop yield performance, and for supporting yield improvement. So Alex has worked at all scales, I would say, of the plant, right from the very small and minute detail of molecular events, all the way through the physiology and all the connections, intermediate traits and so on, all the way up to the whole plant. And he's done this all along with a view of modelling these processes so that we can predict how plants change in different environments and what sort of plants might be more successful than others. So he's generated a priori yield and impact assessment of photosynthesis bioengineering of crops, and of course has impacted fundamental research, fundamental findings as well. And because of all this, he's been awarded an ARC DECRA and a prestigious Australian Society of Plant Scientists Peter Goldacre Medal, which he was awarded in 2020. And he remains at UQ as a UQ Amplifier Fellow. And I'm really looking forward to your presentation, Alex. Before we start, though, I'll just remind everybody that please could you put questions in the chat, and then we'll review them at the end and attempt to answer all the questions. And so I'll leave it over to you, Alex.
Okay, thanks, Christine, for the introduction. Share screen. Okay. Okay, I hope this is coming through. Yes, so thank you everybody for attending this morning. And thank you, Christine, for the introduction. And like to begin with, also like to acknowledge the traditional owner of this land and pay my respect to their contributions. So as Christine introduced, today I'll be talking about crop modeling, and one particular application of this is to helping us to understand, the role of photosynthesis in crop productivity, and how can we incorporate model simulations and model understanding to squeeze more, nuanced understanding, leading to innovative solutions for crop adaptation. And along my talk, I will be highlighting many of my collaborators. So I haven't put their names here, but they will be mentioned along the way. And I think it's important to reiterate, the challenges that modern agriculture is facing, and that's to do with increasing population, increasing food demand on our food crops, and also climate impact. And what we really need is to be able to accelerate our crop improvement progress. But as everybody would know, there's a few major bottlenecks in terms of doing that from a genetic point of view. So, if you are going to select or manipulate plant traits, is not necessarily straightforward, going from gene molecular level to field crop productivity. There's a lot of biological complexities, in between, which I will go into in my talk. And also, we still need to rely on lengthy crop testing processes that takes years and much resources to demonstrate impact at a broader scale. So there's, I would say a pressing need for research, and both research and researchers that can navigate across molecular to field, or as I call it, cross-scale biological complexities, to help us inform, solutions that are more relevant and have higher impact. And I sort of stole this picture from Charlie Messina's paper. It's a scientific method concept that incorporates the use of modeling in the pursuit of science. And what this can add is it can help us enhance the integrative understanding of how plants work, and be able to, along the way, develop prediction capabilities that helps us to do predictions. And what that can do is it can help us identify solutions with enhanced relevance and impact for crop adaptation, and help seed the next round of experiments and research translation. So this thing, the experiment and the modeling world sort of needs to go hand in hand for this to happen. So in my talk today, I'd like to focus on, this application in photosynthesis research and crop yield improvement research. But I also like to say this is definitely extendable to more plant processes and definitely species. So, I don't think my summary of the past, progress in this field can do any justice. So I haven't put any references here. But, several decades of doing this experiment modeling cycle has helped us, understood, to some extent how crop interact with the environment and how it grows across the crop cycle. And here I've given you like an eco-physiological flavor of our understanding. And of course, there are different flavors of, if you work in different scales of field. And today I'd like to focus on the growth factor of crop. And just before that, I'd like to, this is our current modeling, capability, and this is an example of Apsium sorghum model that captures, the key crop physiological factors in a complex network, and also interacting with nitrogen dynamics from the work of Eric Van Nostrand. And together, this network of physiological traits allows us to predict the trajectories of crop growth, development, and yield throughout time. And Hammer et al. Has, reviewed how this sort of tools is useful in terms of breeding and agronomy. And I think, we think the important thing for that to happen is, the coefficients in the model has to have, robust link to whatever the engineers or the agronomists are trying to change. And this coefficient in the spaghetti diagram, has to interact with other components of the diagram and be able to simulate realistic effect across time and scale. So having that in mind, I want to then move on to, talk about photosynthesis. And because there's a global effort in improving plant photosynthesis at the moment. And we started thinking how do we then apply the crop modeling tools to help enhance this global effort, and that has to come from developing nuanced understanding of the role of photosynthesis in crop growth and yield, and in order to generate solutions or guidance for photosynthesis research. And to put that into a crop model, that would need to have some dissecting of some coefficients in the crop models so that we can get closer to whichever processes the genetic engineers or the breeders are trying to get to, so that we can generate some prediction capabilities. And comparing the different ways of modeling photosynthesis growth in modelings, there's the simple biomass is equal to, well, biomass of the crop is equal to how much radiation it's intercepting multiplied by an efficiency factor that converts radiation into biomass. But we also know from other fields of study that in order for the crop to get biomass, it has to perform photosynthesis at the leaf level, and then you have to look at the whole canopy. So what we've done was bring together the different silos of these prior knowledge on the modeling of photosynthesis into the same framework, and that lends us to this I call it the cross-scale modeling that connects molecular photosynthesis molecular processes all the way to field crop performance, and these are the key publications and my collaborators, including Susanna von Cameron, Graham Farquhar, and Graham Hammer. This involves the upscaling of, we have to think about the upscaling of leaf level photosynthesis model to the canopy level, and we've gone with a sunshade approximation of the canopy, as it provides a very robust approximation of the light environment in the canopy, and we also had to think about the downscaling of crop attributes to help modulate the calculations at the leaf level, and that includes how big is the canopy, what's the nitrogen content in the leaf. Those things comes from the dynamic interactions happening during crop growth and development. So really we have to, this is merging the lower level models, the finer level models with the macro level models. And we have the photosynthesis model calculating photosynthetic rates, informing the crop model, the daily biomass gain or daily biomass gain from CO2 assimilation, and the crop model then informing the photosynthesis model, how much water be available for doing photosynthesis, what's the nitrogen content for the machinery, photosynthetic machineries. So it's a two-way interaction. That's the essence of this cross-scale modeling. And to put this into some action for you, here is a simulation of a crop cycle. Here we have simulating the stages through the crop growth, from sowing to maturity, and as you would expect, as the crop grow, leaf number increases along the time, and when leaf number increases, the leaf area index of the canopy increases, and that's why you start to see a pickup in photosynthesis rate in the early phase of the growth. And as this crop performs photosynthesis, it demands water. So this is your daily water use. And with doing photosynthesis, your biomass growth increase. And then you hit a plateau in your leaf area index of the canopy. So you have a plateau in photosynthesis. So you sort of get the overall picture of, the photosynthesis calculations is informing the growth of the crop and vice versa. So it's quite complex. But what we've done was we were able to test the robustness of this cross-scale modeling framework. So here I'm just plotting out all the observed yield in previous experiments and the predicted yield using this cross-scale model. As you can see, it follows, it correlates with each other very well, indicating the robustness of this cross-scale modeling framework. And by doing the cross-scale modeling, we were able to unlock new prediction capabilities, because now we can then start to ask questions about what happens if we change, for example, the rubusco or the electron transport bit of the photosynthesis, which I'll go into later. But here I also like to make a very important point about model development or model advances. So one of the things it was really nice that we could maintain the robustness of our model simulations or model predictions. Here is the comparable plot, but just using the radiation, the RUE approach of calculating canopy or calculating biomass growth. And this is showing us, by extending the FCM sorghum framework to include a lot of details about photosynthesis and canopy interactions, we haven't reduced the robustness of the model. So this is a very important thing in terms of model development. And so with this cross-scale modeling innovation, I think this allows us to start getting a priori assessment of photosynthetic manipulations. As I said, there's a global effort in improving plant photosynthesis, and any nuanced understanding, can potentially lead to higher relevance and impact with regards to photosynthesis research. So I've teamed up with a bunch of people in the Center of Excellence for Translational Photosynthesis in trying to answer some of these questions about the impact and relevance of manipulating photosynthesis. And by the way, the cross-scale model, we developed it for both sorghum and wheat. In this talk, I'm mainly focused on sorghum simulations and results. So we've sort of summarized the photosynthetic manipulation into 9 strategies by talking and working with this group of people for sorghum. So this is another cross-scale model in action, and I like to show this as a first simple case for you to understand what this is trying to do. So I think I've shown you, as the crop grow over the season, your biomass increases. So that's what this line is. And with the operation of the cross-scale model, this is driven by the canopy doing photosynthesis. So every bar here is the total canopy photosynthesis for the day, and that accumulates to give you this biomass growth. Now this comes from parameterizing your photosynthesis versus CO2 response curve. So if you have your model parameterized for this response curves, we can then simulate canopy photosynthesis at every conditions and across every days. So with manipulation, so assuming that the baseline photosynthesis response is the black curve, with manipulation, it increases let's say it increases the curve to these colored ones. Now, what this would likely do is here it would increase your biomass gain over the season or over some time, and that's because it's driven by higher canopy photosynthesis rate. And there's some more interactions later in the season, which I'll go into later. But the key thing here is when we input this increased photosynthesis at the leaf level, it's telling us that yes, photosynthesis of the canopy goes up. Here is about, for the sunlit leaves, is about 10% increase, and that drives the 10% increase in biomass. So it's sort of working in a way that we expect. And this is the fuller picture of all the simulation that sort of happens in the background. So with this capability, we play around with simulation studies to generate some a priori understanding of photosynthetic manipulation strategies and what they mean in terms of, for example, this particular target environment, target population environment, the northeast of Australian sorghum growing region. And this study is, you can sort of think of it as an in silico yield trials, that if you were to do it in reality is probably not feasible, because you need to generate the 9 photosynthesis manipulations. You need to test them across 4 growing sites and across 120 seasons, different seasons, with different starting soil water conditions and different sowing dates. But this is quite, easily achievable with this modeling framework. So that's what we've done. I haven't really introduced what these numbers are, but they're just different ways of manipulating photosynthesis from 1 to 9. With irrigation, we predicted that one of the, a couple of the manipulation strategies could potentially give us a 10% increase in sorghum yield, which is very big. But the reality is usually not something different. When we switch off the irrigation in the simulation to just do dry land cropping conditions, what we saw was the yield increase dropped significantly from about 10% to 4%, and then you see a lot of variation that's coming from the different seasons interacting with different conditions, starting conditions. So I guess the key takeaway from this study, this major study, is that photosynthesis manipulation is probably not likely to give us a step change in crop yield. But this leaves us to think about, okay, so what else can we do to enhance the relevance and the impact of photosynthesis research? And I'm in the process of developing major funding opportunities to pursue those avenues. So hopefully by the next time round, I have something to say about that. But so this large scale simulation gives us 2 things. It gives us testable hypotheses for further manipulation, experimentations. And it also generates a very large data set that people can utilize or exploit to understand the interactions across scales and what's driving yield, or why isn't it driving yield. And I want to give you one example of that. So we went back to look at our daily canopy photosynthesis simulation across the season. And we've noticed that for a majority of the time of the period, it's dominated by this red shading bars. Now this red shading bar says that the canopy photosynthesis is limited by light driven processes or electron transport limitations. So we were thinking, okay, what does this really mean? Because it's like a really big chunk of your crop growth cycle. So if we can somehow do something to this bit, maybe there's maybe it's more relevant and has more impact. And that's why we, started looking at some experimentation and further modeling, which we recently published in New Phytologist. And the details about this study is that okay, we got some understanding, some ideas about electron transport limiting canopy photosynthesis and growth. We also have from other disciplines, we also know about different crops have different radiation use efficiency. So that's crop per solar radiation intercepted. And we were thinking maybe the two could be connected, and that's something we could test out via this cross-scale modeling framework. So we were asking the question: is there any potential link between crop level radiation use efficiency and leaf level light electron transport processes? And this is the motivation for us. So maize has a higher RUE, sorghum has a lower RUE. They're both C4 species. Perhaps there's something in the photosynthesis. So we devised a pairwise method of measuring photosynthetic responses to both CO2 and light. So these curves will measure, we select leaves and then we measure these curves, consecutively, one after the other. And what this allows us to do is to improve the parameterization of photosynthesis to physiological responses, and help us to parameterize the model, so we can calculate photosynthesis under all combinations of conditions. And this is different from what's typically done in the discipline, where they look at CO2 response separately to the light response. But here we're arguing that we should put them together so that we can parameterize our models better. And I went ahead and collected a bunch of data from my glasshouse trials, running light core machines, looking at two plants. So your hybrid sorghum and then a hybrid maize genotype. And I measured a bunch of CO2 response and light response in the pairwise manner across many temperatures and collected many reps. I think I ended up spending about 100 or 200 hours of light core machines, which was quite a substantive measurement. But this allows us to then extract C4 photosynthesis model parameter values from these data quite robustly. And we were able to look at the maximum Robisco activity response to temperature, that's here, and also Pep-C activity to response to temperature. We also had inferred the maximum electron transport rate from fitting these data, and also the fraction of PAR observed by photosystem 2. So it's a parameter that sort of influences the initial bit of this light response. And Sandra and Ryan from Corteva, they were instrumental in helping us develop the Bayesian model fitting approach, which allows us to sample a large number of credible fits. So we were hoping to get, all the credible fits so that we can look at look at genotypic differences. Here I've shaded the green bit, and that's just to highlight this sort of temperature range is the what's relevant for sorghum and C4 crop growth. And the major difference in this parameter in the PR absorbance of PS2 happens around relevant growing temperatures. So we've then put all those parameters into the model, into our canopy photosynthesis cross-scale model, and we simulated what happened to a canopy under a sunny day in the middle of the day. So we calculated the light levels at different layers of the canopy, and then we apply the photosynthesis parameters modeling to calculate what's the photosynthesis performance for each layer of the canopy. And the black and the yellow contrast is the performance of when we use the sorghum photosynthesis parameters, and the yellow is when we use the maize photosynthesis parameters, which, as you can see, performs better than the sorghum under the same light conditions across the layers. And what this is saying is that it is likely that the performance of photosynthesis under non-saturating light could be a key for canopy photosynthesis and RUE. So we extended this modeling to simulate across the day. So across the day, you have variations in PAR or light, variations in temperature. And this is our simulated predicted canopy photosynthesis with sorghum photosynthesis or sorghum crop with maize photosynthesis. That's the added black bars. And when we calculated the final outcomes for RUE, it's telling us that if we were to change this sorghum photosynthesis to use all the maize parameters, we could potentially increase radiation use efficiency by 20%. So that's a very big increase in this such important adaptive trait. So what this tells us is that, okay, this could be a useful, relevant, impactful, trait that further breeding selection or manipulation experimentations could follow up with. And I also want to move to another example, which I think hopefully I have still more time to talk about other examples, but we'll see. The other example is that when we are digging through our large scale data sets, I talked about this red shading being the light limitation and how that leads to our study in new phytologists, and finding out the low light response is key for driving growth. There's also other aspects that we identified from our data sets, is that we noticed that when we increased our photosynthesis in our simulations, it's coupled with increased stomata conductance. So stomata conductance on this ACI plot is basically this slope here. It's photosynthesis, so that's your y-axis divided by your change in CO2 x-axis. So this change in CO2 comes from the difference between air CO2 and what's inside the leaf. So that's your delta CO2. So on this graph, you can see that if we move our photosynthesis rate vertically up, this slope is going to go up. So conductance is going to go up. And what this sort of resulted in is that, okay, yes, photosynthesis goes up, like I mentioned in the beginning, in the previous example. Your canopy photosynthesis performs here about 10 less than, just about 10% better than your baseline simulation. But what happens with this increased conductance is that as your crop grows through the season, it demands more water, but your soil water is running out. So that's why you have this empty space in these bars. They are the demands not met by water supply to the crop. So what happens with photosynthesis enhancement is that, yes, it increases biomass, increases photosynthesis, increases biomass, but it also increases water demand by the crop. So you run into a bit more severe water stress. So this is your supply over demand ratio. The lower the number, the more water stress the crop gets. So with enhancement, you get more water stress, and this impacts your late season performance. So your crop, your grain field and photosynthesis during that time. So we teamed up with Andrew Leakey from Illinois to explore different ways of try to mitigate, try to find solutions to mitigate this problem. And we've thought about a few hypothetical situations involving increasing photosynthesis and combining with decreasing stomata conductance. So it's changing the ACI curve up and down, but also trying to change the slope to reduce the conductance. And here is a simple simulation of a sorghum crop at one site over 120 seasons, different, try to get the seasonal variability. That's why you get lots of points across this x-axis. So here are three scenarios that we tested. Singly enhancing photosynthesis, that means you also have increase in conductance that comes with it. So that's going up. And we also tested, okay, what if we just reduce conductance? So we're falling down the ACI curve and something in between. And what this tells us is that under this particular target environment, it looks like reducing conductance alone is a very impactful, very relevant approach. And that, this study got us thinking. So one of my PhD students, Xia Jin, we've then started to look at, okay, what's the relevance of reducing conductance, increasing photosynthesis, or a combination of them mean in terms of plant level or crop level performance? And we measured. So we've started measuring a lot of stomata conductance response to environment and photosynthesis response to environmental conditions. And what we're trying to do is we want to then be able to parameterize the model and use that to explain any linkages between leaf level performance and whole plant level performance in terms of transpiration efficiency, which is one of the very, very important adaptive trait for water limited environments. And the motivation of doing this is, if you talk to crop physiologists, breeders, knows the importance of transpiration efficiency, and molecular physiologists knows the importance of the trade-offs between net photosynthesis and stomata conductance. What we're trying to do is we're trying to develop an integrative understanding of how the two sides are linked together, and perhaps helping us to identify nuanced photosynthesis and conductance understanding and the trait understanding and see what sort of combinations could we be targeting at the leaf level to enhance plant level transpiration efficiency. And to do this, my student and I will move into looking at perhaps first establish the science of the linkage between the scales and identify key traits, and then developing high throughput phenotyping methods of targeting these key traits at the leaf level, which we anticipate have some linkages to the whole plant level performance. And that doing this, could potentially provide us new ways of targeting plant water use efficiency. And I just want to mention this, interesting study Andrew Leakey from Illinois. So they've sort of pursued after, well, perhaps also inspired by the simulations that we've done together. They've went ahead and bioengineered a sorghum model sorghum genotype to reduce stomata density. So this is the bioengineering pathway of looking at applying the simulation results. They've showed that in their sorghums, they reduced conductance, impacted photosynthesis a bit, but overall leaf level water use efficiency went up. And they were tracking, the behavior of these plants across some period, dry down experiment, and they showed differences in water usage per plant. They showed a crossover in stomata conductance between the wild type and the engineered plants. The wild type crashes after some days of some days of dry down, but the engineered ones with reduced conductance kept on going. So there's a lot of interesting results from this paper that is currently they put up on the web. But I think I would like to say, this provides us another stepping stone into further modeling the engineering of the plants, because I expect in doing that we can further enhance our understanding of what's actually being created. We can assess their relevance and impact across target population and environments. So again, I argue this has to go in circles between experiment and models. So before we arrive at a robust solution, we need to keep doing this sort of scientific exploration. And just lastly, I want to finish up with some other model development that I'm doing with my other PhD student, Genevieve Darrington. And this is to enhance our crop simulation models with the ability to sample a large space of the crop adaptation landscape. So imagine that there's countless possibilities of combinations of genetics, perturbations, and management perturbations. We want to be able to quickly sample through the combinations and identify optimal solutions for crop adaptation. And this is what this capability is trying to unlock. And we ran some initial testings of the framework by looking at optimizing yield per transpiration, evapotranspiration allowance, and we can see the framework or the algorithm taking us from these very undesirable combinations of crop designs in terms of tiller number and maturity length to these more optimal designs. So this is currently under preparation, and we hoping to submit this very soon. And the plan with this is, okay, we want to start looking at the growth traits, photosynthesis, conductance trait, and also combining future climate conditions. And we expect this sort of tools to generate, a lot of further testable hypotheses for crop designs to seed next rounds of experiments. So this is my last slide. So what I've said during this talk is that through the cross-scale modeling between photosynthesis and whole crop performance, we have enhanced the understanding of leaf photosynthesis, the role of leaf photosynthesis in crop growth and yield. And we've also developed a prediction tools that enables us to, assess the impact of photosynthesis, the impact and relevance of photosynthesis. And we've generated informations that can inform cross-scale strategies for photosynthesis improvement. And lastly, I'd like to just end with this statement that translational research requires funding that rewards a balanced fundamental research, field research, and importantly, research that look at the integrative understanding and prediction tools, capabilities to help us enhance the relevance and impact. And like to end here. So thanks very much for listening and happy for questions.
Thank you, Alex. Fantastic presentation. Really enjoyed it. There's quite a lot of questions on the chat, which I can now go through.
Okay.
We might just shut down your slide sharing just so that we can see you more easily and answer the questions. Hear your answer for the questions. So first one's from Michael Advardi, who's asking. He's basically asking whether these high CO2 experiments show a balance against nitrogen. So where high CO2 experiments might reduce the nitrogen quality. Is that the case? And can modelling help mitigate this trade-off?
So we saw in our simulations that with the same amount of nitrogen uptake, if you have more carbon in the system in your crop, now obviously the nitrogen per carbon is going to go down. So we can see that in our simulations. Now, can modeling help to mitigate? I think that's a yes. So what we need to do is we need to understand what's limiting your protein or nitrogen content through simulations and design experiments to test mechanisms or hypotheses so that we can help retain or maintain your nitrogen content.
Okay, I'll just ask, a few people have asked questions. If you want to ask the question directly, just turn your video on and then I'll know that you want to ask the question directly. But if I just jump next to Alex Norton. Oh, Alex, you're there. Would you like to ask your question directly to?
Yeah, that'd be great. Can you hear me?
Yes, I can.
Great. Yeah, really nice talk. It's nice to hear all the work you've been doing. Just thinking about these seasonal simulations that you showed, particularly thinking about, the sort of the model outcome that there's like a light limited photosynthetic rate for the first half of the season. That's sort of what it looked like. The kind of time scale of measurements that you would do from a LICOR instrument is very different to what you would see across a season. So I'm wondering, in your actual model, how does Jmax and VCmax, the optimum of those at least, change over the season? And is that change linked to maybe nitrogen for VCmax or something else for Jmax?
Okay, so our our modeling of Jmax, Vcmax across the season is based on observations where people looked at measured Jmax and Vcmax and also leaf nitrogen content. And they saw quite a robust response between certain nitrogen content. So what we've then done is in the model, we then linked Vcmax, Jmax at 25 degrees to leaf nitrogen content. And this nitrogen content in the leaf actually changes through the season, because in the beginning you might have high leaf nitrogen content, but when your grain starts to set, you have a lot of nitrogen sink by the grain. So, some of your leaves might senesce or reduce in nitrogen content. And that's when you start to take a hit in your Vcmax and Jmax values at 25 degrees. So typically later in the season. So, through this Vcmax nitrogen relationship, we can capture how it may vary across the season.
Yeah, great. Yeah, that's a good explanation.
Thank you. So Ken Webster would like to say hello, and does the biomass growth incorporate cellular maintenance costs?
Yes, yes, we calculate gross canopy photosynthesis, and then we take off a fraction to account for maintenance costs. Yep.
And that's a fixed fraction as a proportion of the biomass, is it?
That's a fixed fraction of assimilated CO2 at the moment in the model. Yeah.
Okay. Okay. Tony Fisher asked a couple of questions. How does the detailed version of the model handle crop respiration?
So just like I said, so we calculate total canopy photosynthesis across a day, and then we take off a fraction to account for that.
Yeah, same sort of fraction thing. I'll just ask Tony's second question. The final example looks a bit like the transpiration limitation rate, which I believe in May has delivered the Corteva Aquamax hybrids that indeed our chief investigator Mark Cooper was involved in developing. So you probably know a bit about that.
Yeah, I don't know if yeah, maybe Mark is there. Correct me if I'm wrong, but I don't know if the photosynthesis conductance aspects were looked at in the Aquamax hybrids. Sorry, I don't know about that.
Yeah, he was talking about the transpiration, the question related to the transpiration limitation trait. And I think, yeah, certainly it was related. Yeah, his question was about transpiration limitation.
Yeah, I mean, it could be linked.
Yeah.
So, yeah.
No worries. Paul, did you want to ask a question?
Yes, indeed. Thank you. Yeah, Alex. In one of the last graph, you shows the lower yield for I think it was sorghum that was transpiring more, right? So you have a higher transpiration, but a lower yield. Do you have any idea of what the limitation is there? Because I mean, you talk about carbon, but could there be any other limitation like nitrogen? Because you will expect that higher transpiration should normally bring more nitrogen to the leaf, but since it's probably done, this measurement were probably done with a constant nitrogen in the soil that could, there's maybe less access to nitrogen or something else limiting. So do you know what the limiting factors are for the ones that are transpiring less, more, sorry?
Yeah, so how it plays out in the simulated dynamics is that when we enhance photosynthesis in the model, it also increases stomata conductance and obviously transpiration. So which means your crop would be using more water compared to the baseline, and that drives the soil water down quicker than the baseline crop. So you have a bit less water during your important flowering and grain filling period, and that additional loss of water during that period means it has less capacity to do photosynthesis, to get carbon. So that's why you can end up with less yield with enhanced photosynthesis when you also have water limitation later in the season.
I'd like to ask a general question. Right at the beginning, you said that when you built all this sort of photosynthesis aspect of the model, and then you connected it into AppSim, you had to check that it didn't sort of, accidentally lead to a poorer prediction from the AppSim model. And the point there is that adding more detail to models often does diminish the accuracy of the model. So I mean, I'm just wondering if you could sort of speculate a little bit about that. Why does that happen, and why did it not happen in your case?
Yeah, so it can happen if you're going into a bit more details in the model, and you're assuming some parameters, could be from just point measurements or some point in time, and then you're then using that measurements to go across the whole season, and which might not be realistic. And this actually comes back to Alex Norton's question, quite, is relevant. So if we were to parameterize photosynthesis ACI curve for the crop the same way across the whole season, and we don't change that and just maintain it the whole way, I think our simulation would be pretty bad.
Yeah.
But the fact that we built in, physiological understanding of how we can, use the model architecture to calculate how photosynthesis actually goes up and down across the season, that I think helps the simulation, quite well.
Yeah.
So the knowledge that's coming from the crop level model is robust enough that when we downscale it to the photosynthesis level, it's still sensible. I think that's pretty important. But if you were to just increase the complexity of the model without thinking about how to parameterize the thing, and just use some default parameter across all environment, all time, then I think it's quite dangerous.
Yeah, agreed. Hey, well, that was a great list of questions and a wonderful talk. Thank you so much for talking with us today, Alex. Really appreciate it, and it was really, really useful. So thank you. I remind everybody that there will be a recording. So if you want to listen again to anything that Alex has said, you'll be able to get it on our YouTube site. Or if there's colleagues that you think might like to have heard this talk, and you'll be able to share the site with them. So I hope everybody enjoys the rest of their day, and thank you again, Alex.
Thanks, everybody.
Thanks. Bye.